Welcome, everyone. Thank you for joining us for today's keynote presentation titled Discovery and Development of a Novel First-in-Class Resuscitative Agent. It's my pleasure to introduce to you Professor Anil Gulati. Professor Gulati is Chairman and the Chief Executive Officer of Pharmazz Incorporated and is an adjunct professor at the Colleges of Pharmacy and Graduate Studies, Midwestern University, and Department of Bioengineering at University of Illinois at Chicago, Illinois. For a complete biography on our speaker, please visit the Biography tab located at the top of your screen. My name is Xavier Gutierrez, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. And this presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Professor Anil Gulati. I will now turn the presentation over to him.
Hello, everyone. This is Anil Gulati, and actually I'll be talking about the discovery and development of a novel first-in-class resuscitative agent. And so I'll give you a brief how we discovered it, how we are developing it, and how we are planning to take it all the way to the market. So basically, this product is for massive blood loss. And blood loss mainly takes place from as a result of trauma, postpartum hemorrhage, GI bleeding, complicated surgeries, drugs like aspirin and others. They can cause the massive blood loss. And there is, whenever there is blood loss, the first thing medical attention is needed is to control the loss of blood and then support ventilation and cardiovascular function. And many times these are intensive efforts, and there is a significant unmet medical need. Although there is very extensive research which has been carried out, but mortality still remains very high, and there is almost like 20% or 25% mortality, and some of the developing countries it's close to 30-35% mortality in such cases. So there are two types of blood loss or hemorrhagic shock, which are controlled hemorrhagic shock, where the source of bleeding is occluded. These are easy to handle, but most of the time what we come across is uncontrolled hemorrhagic shock. Bleeding is temporarily stopped, and most of the time it is because of hypotension that blood pressure goes down, so the pressure in the arteries is, and the clot forms and tries to stop. But sometimes the problem is when we reperfuse, the clot dislodges and once again the bleeding occurs. So what we try to do is we try to restore the pulse, try to restore sensorium, and the phenomena of hypotensive resuscitation, that is obtaining blood pressure of about 80 millimeters mercury. So because of this unmet medical need, nothing much has been discovered over the last 40 years. All we use is vasopressors, and there is vasopressin has been used, and recently angiotensin II has been approved by FDA. But really nothing major has happened excepting use of vasopressors. The problem in vasopressors is that they constrict the arteries. If the arteries are constricted, the blood flow will be impaired. Although the blood pressure is going up, but still the blood flow is likely to be impaired. So this compound, centhaquin, this in our animal models of rat model of fixed pressure blood loss, rabbit model of uncontrolled bleeding with trauma, a pig model of massive blood loss. In most of these different models, we found that it reduced mortality, It decreased blood lactate levels and increased arterial pressure, pulse pressure, and most importantly, cardiac output increased. And we also went ahead because this was a new molecule and never being any scientific or international non-proprietary name was not there. So we approached WHO and got the name approved, Centhaquine with an E. That has been approved now. That is the official name for this compound. So this tracing is the first one in which we, first experiment which we did in which we found that Vickers model where we use the blood pressure on the top two panels are the blood pressure, bottom is the heart rate. And you can see that the blood pressure drops down to 35 millimeter mercury, and we keep it for 30 minutes. We keep it at 35 for 30 minutes, and then we resuscitate. And at 30 minutes, when we resuscitate with Centhaquine, we can see that the blood pressure is going up. But surprisingly, the heart rate is going down. And most of the vasopressors which are used today, the heart rate goes up. And so this was a surprising and novel finding which we took into account and expanded our studies to measure the left ventricular pressure, left ventricular volume, in which you can see the loops are generated. And the loops, the diameter or the width tells us the stroke volume. That is of important thing which we should know. And the height tells us the pressure. And one in the sham, these pressure volume loops, we can see that there is not much of change. But when the blood loss occurs, we can see that pressure has dropped and the width also has dropped. So the stroke volume has reduced significantly. When we resuscitate with 3% saline in the middle panel, we see there is some resuscitation or some improvement in stroke volume and pressure, but it is transient. And by 240 minutes, none of the animals survived and all of them died. Whereas in Centhaquine, you can see that the pulse pressure, the left ventricular pressure and the stroke volume are significantly improved. And 240 minutes also, there is good survival. So this is an example of a pig study which was done in Greece by Theodore Xanthos' group. And so where the severely hemorrhaged pigs, almost 50% of blood was lost, and then they were resuscitated with either saline or with Centhaquine plus saline. And we can see to bring the blood pressure back to baseline. And we can see that when we needed close to 2.2 liters of fluid to bring the blood pressure back to baseline, and when we added Centhaquine, the blood pressure was only, the blood volume which was needed, or the volume of fluid which was needed, was only 300. So after 2 hours of experiment, there were only 2 out of 10 animals survived in the control group, and all 10 out of 10 survived in Centhaquine group. 24 hours later, 0, none survived in the control group, and 9 out of 10 survived in Centhaquine. So this was a good indicator of survival. Now this is from the same swine study in which 50% blood was lost, and we can see on the top panel is the cardiac output and bottom is systemic vascular resistance. The red one is the control and black is Centhaquine. You can see a significant increase in cardiac output, which has taken place in Centhaquine. What happens to vascular resistance? It goes down. Most of the vasopressors which are used today, they increase vascular resistance, and this one uniquely decreased vascular resistance. So that is something very, very important. And then further studies were carried out in another group of pig model by Theodore. And we found that wet-dry ratio or the lung capillary permeability was significantly improved. Horowitz index significantly improved, so that the lung function of pulmonary edema is less likely to happen. Mortality also was significantly improved. The unique approach of this is it has the potential to be a first-in-class therapy. The reason is there is no compound which is increasing cardiac output and decreasing vascular resistance in hypovolemic shock. This is a unique thing, which is so a unique way of treating patients with massive. Mechanism of action, how does it act? There are a lot of receptors present. Alpha 2B receptors are on the venous side, and they increase mean circulatory filling pressure. and that leads to an increase in blood pressure. Simultaneously, in central nervous system, there are alpha-2A, which are sympatholytic, and they will decrease vascular resistance and decrease blood pressure. Net effect of these two is an increase in blood pressure in hypovolemic shock. That means the venous part is predominating. So what is happening is Centhaquine stimulates alpha-2B venous receptors, which increase venous return to the heart, increase cardiac output, heart pumps more blood per beat and improves tissue perfusion, while simultaneously it acts on alpha-2A to decrease sympathetic drive. Blood vessels are dilated, or the arterial side is dilated, and the blood flows easily through the tissue, through the vessels to the tissue. So 70% of our blood is pooled on the venous side, which is effectively not circulating or supplying oxygen and nutrition. 30% arterial side is the one which is supplying. Centhaquine, because of its venous alpha-2B receptors, moves that blood towards the arterial side and sustains life. So, and veins are very compliant. They can accommodate a large amount of blood, and that's why almost 60-70% of blood is pooled on the venous side. So this was the basic mechanism which I just elucidated, but now I'll give you some of the clinical data. We embarked on a clinical phase 1 study, which was done in this center. And what we did was we did single ascending dose, multiple ascending dose. The single ascending dose was 4 groups, and multiple ascending dose was in 2 groups. So our selected dose for hypovolemic shock is 0.01. So we 5 times higher, there was no adverse effect. 10 times higher, there was no. 15-fold higher, that's the time when we saw the adverse effect. Multiple ascending dose, there was no adverse event report. So in other words, the safety is so high that we can go even 10-fold higher and no significant adverse effects were observed. So once again, in ICU setting, most of the drugs which we use, particularly vasopressors and others which we use, you increase the dose and chances of adverse effects are there. So safety is very, very big. And adverse effects are also very, they are expected. For example, hypotension was observed when 15-fold higher dose was used. And this was expected because of alpha-2A activity in the CNS, which is causing a fall in blood pressure. So that dominated and caused fall in blood pressure. And this lasted for 1 hour, 5 minutes in this patient and or subject and resolved by itself. No medical intervention was needed for any of the adverse. Other minor adverse effects like drowsiness and all that, dry mouth, are expected because clonidine-like action. So no major adverse effect was observed in. So we have established the safety, and maximum tolerated dose was found to be 0.1 milligram per kilogram. Well tolerated, both single ascending dose and multiple ascending dose. Proposed dose is 0.01 milligram per kilogram, which is 10-fold or 10 times lower than the maximum tolerated. It has a short half-life with large volume of distribution. So basically, once the phase 1 was established that it is safe, and so we embarked on human phase 2 studies. And so it was a prospective, multicentric, randomized, blinded, parallel, saline-controlled phase 2 for excessive blood loss. All the subjects received standard treatment for shock. So whichever is the standard treatment, which was to be as soon as the patient arrived, and when the systolic blood pressure became less than 90, standard protocol resuscitation kicked in, and that's how the patients were treated. So one arm was control, where standard treatment was given, and the second arm was Centhaquine of TMZ 2010, where they received the standard treatment. In addition, they got Centhaquine. These are some of the centers, 7 centers in India, where the trial was done. So 137 patients were screened and 50 were enrolled. So out of 50 enrolled, 22 in the control completed the study and 23 in Centhaquine completed the study. So we will present the data on those demographics. You can see that the control as well as centhaquin, the gender is very similar, age is similar between the two groups, body weight is also similar, height, BMI, and body surface area, both are similar. So basically telling us that we are comparing apple to apple, and these groups are comparable to each other. Further, the number of doses were to be given in 48 hours also were similar between the two groups, and statistically they were not different. Now, the important thing is whether the blood loss was similar or not. So hematocrit level. So we can see that hematocrit levels also in both groups are very, very similar. So the blood loss also in both the groups are similar to each other. So once again, emphasizing once again that these are comparable groups and they are similar to each other. Injury scoring value. This is also an important marker. So some patients, it could have been argued that one group may be having more injury and second be having that. But here we can see that injury scoring value is similar to each other. P value is 0.56. So that means they are not different. Fluids infused over the first 48 hours are also similar. They are not different. Blood products like platelets or RBCs or blood, when they were transfused, they were also similar between the two groups. So both groups received similar amount of fluid in first 48 hours and similar blood products in the first 48 hours. Number of patients on vasopressors or the patients needing vasopressors, it was about 41% in standard treatment and 26% in. So there is a trend that there is a decrease in number of patients requiring vasopressors when we use centhaquin. Now, this was not statistically significant, but there is a clear trend. And the amount of vasopressors which are to be infused, which were infused in first 48 hours, is 9 compared to 3. So from 9 it dropped to 3, but standard errors are quite high. So there is a trend that less number of patients are requiring vasopressors. The amount of vasopressors needed in this group is less, but statistically it has not come out to be significant. So the percentage time in hospital, how much of time was spent in ICU also is similar. There is a trend that there is some decrease in ICU stay, but predominantly it is statistically not significant. So time on ventilator from about 2 days down to 1 day, but errors are high. So there is a trend that there is decrease in time spent on ventilator, but statistically it did not come. Serum creatinine values. So we can see that both control and centhaquin group, they are showing improvement in serum creatinine. So P value is 0.065 control and 0.0009 in centhaquin. So this level of significance is a little higher in centhaquin group. But this tells us, all the story is telling us that the control or standard treatment also is working. And that is a very, very important part in a clinical trial, that the standard treatment should be working. And here patients are recovering due to standard treatment also. Glomerular filtration rate, very similar to serum creatinine, so 0.003 in control and 0.0006 in centhaquin. Now comes the important clinical biomarkers, which are blood pressure, base deficit, blood lactate levels. And I will go through these clinical biomarkers. We can see that the red is the standard treatment and green is standard treatment plus centhaquin. So we can see that the blood pressure is very clearly better in centhaquin group compared to standard. And when we do individual patient analysis here in this, so we can see that the systolic blood pressure, when we compare 0 to 12 hours or 0 to 24, 0 to 48 hours, they reach barely significance. And in some cases over 12 hours, they did not even reach level of. Whereas in centhaquin group, highly significant, less than 0.0001 in all cases, whether it is 0 to 12, 0 to 24, or 0 to 48 hours. Diastolic pressure, the separation is a little bit more clearer. And when we do individual patient analysis, then do analysis of variance, find that the p-value did not reach the level of significance in the control group, but in centhaquin group, it is 0.0001 and less. So highly significant improvement in diastolic. So base deficit, which has improved in both the cases, but in centhaquin group, it became base excess. So individual patient analysis, there is improvement in both, but the level of improvement or the p-value changes. Blood lactate level, which is one of the most important indicator of mortality, significantly decreased in control from 4 to 3 and from 4 to 1.4 in centhaquin. So highly significant decrease in centhaquin group, but in control there was a minimal decrease. So blood lactate level, when we compare individual patient analysis, it is showing that there are from 0 to 3 days, the p-value is 0.29. It did not reach the level of significance, whereas in centhaquin group, it reached highly significant decrease in blood lactate. So here there are two patients which I want to emphasize, patient 10 in control and patient 12 in the treated group. So this patient, these two patients, 005 and 006, these are the two subjects. So 005 is the control, saline, and 006 is the centhaquin. You can see that both are females. Both had pregnancy which went wrong. And one is 24 years age and the other is 31 years of age. One had 005, which is the saline group, had baseline hemoglobin level of 3, while the patient in centhaquin group had a hemoglobin of 3.6. And the patient in saline group died, and the patient in centhaquin group survived. And lactate level in the saline group patient, that was 005, was 11.18, and it went to 15.3, and the patient died within the first day. While the patient on centhaquin group had lactate level of 17.41, and on third day, the patient had lactate level of 2.4, and this patient. So there were total mortality, there were 2 deaths in the control group and none deaths in the centhaquin. So adverse events, there were 3 adverse events reported in control and 2 in centhaquin group. And these events were not related to the drug, because the acute kidney injury, one of the adverse events in acute kidney injury, was in a patient with a stab wound where the knife went all the way to the kidney and damaged the kidney also. So the kidney injury was because of the wound, or because of the stab, and not because of the drug. So how it is given, we dissolve the contents in a vial, which is 10 ml of normal saline, and we reconstitute 0.1 milligram of centhaquin, which becomes 0.1 milligram per ml. And then supposing if the body weight is 70 kilogram, we take 7 ml of this and reconstitute in 100 ml of normal saline in the bag and infuse over 1 hour. So it is a very simple procedure. So while we put 10 ml of saline, dissolve it in 5 to 10 seconds, it dissolves, and then we take according to body weight, whatever amount, suspend in 100 ml bag, infuse over 1. So how does it act, pathophysiology? So centhaquin, as we know, that it increases venous pressure when we infuse, and that increases venous return, ventricular filling goes up, ventricular stretch goes up. Frank-Starling's law, more ventricular filling, more will be pumped out. So ventricular contraction increase, stroke volume increases, and that leads to increase in cardiac output. Simultaneously, centhaquin also, I showed you in the first slide, that it decreases the heart rate. If the heart rate decreases, then the diastolic time decreases. And diastolic time decrease, that will allow more of filling time, or diastolic time will increase and more of filling time will increase. So more time now for the blood to fill into the left ventricle, that increases left ventricular filling, which increases ventricular stretch and cardiac output. Now, when we are infusing fluids and blood, and they are also increasing the blood volume. So all that combination, when fluids and blood volume, fluids are infused, they are helping increase the blood volume. Filling time increasing, that is increasing the ventricular filling. Venous pressure is increasing the ventricular filling. So all that leads to an improvement in cardiac output, and that leads to, and simultaneously we know that this increase in cardiac output has to go and supply the tissues and organs and blood vessels on the arterial side. They get dilated because of centhaquin. That leads to an improved perfusion. So that is the pathophysiological mechanism by which. So compared to present vasopressors, Centhaquine or lifaquine, there is no evidence of tolerance. So vasopressors, they do develop tolerance. The vasopressors presently use the increased heart rate. Here it decreases heart rate. Then vascular resistance increases. Here it decreases. The increase in cardiac output is because of increase in heart rate. Here increase in cardiac output is because of venous return. There are beta receptors involved which can lead to arrhythmias. Here there are no beta receptors involved. We have conclusively shown that no beta receptors, no arrhythmia. So isomers are present in the vasopressors, particularly dobutamine, lifaquine, no isomers. There is evidence of fluid extravasation. There is no evidence of fluid extravasation here. So the goals of resuscitation, like restoring cardiac function, improving end organ perfusion, reversing metabolic process, coagulopathy, and output, all the goals are achieved. And so this is basically a safe and a highly effective first-in-class resuscitative agent. And basically, a lot of value additions are being added to the presently used suppressors of the treatment of shock, hemorrhagic shock, by highly safe, 10 times safety, highly effective, reduction in mortality, innovative, first-in-class medicine, and society benefit, unmet threatening, life-threatening conditions, so, and reducing the adverse effect. So there are limitations at present with the subject population, although it is multicentric, but it was done only in one country, not a global trial. We are now embarking on a global trial. We met with US FDA, and there are positive feedback which we got. And since it is Frank Starling's law, we are recommending that decompensation, it may not be useful. And then it is right now limited to hypovolemic shock, but there are, it could be, we are doing experiments in animal models of septic shock, see whether it can be used in or not. Now, we have, although I'm not presenting the data, but we have already done phase 3 trials also, and we got similar finding to what we got in phase 2. And that phase 3 trial was done in 105 patients. And that trial data has been presented to US FDA. And we have also applied for marketing permission in India. So the next few months, we do expect that we should be able to get some positive responses from the regulatory agencies. So this is an example of, in the end, I thank you. And if any questions, please do not hesitate to email me. Or so this is an example of how in our academic institution at Midwestern University, when I was working as a dean of research and professor there, we discovered this compound and found out a new use of this compound. And then university protected it intellectually. Intellectual properties, and it was licensed to the company which I founded in 2010. And now it has come to the stage where it is about to come out in market, and in US and other countries on a global basis. We are likely to embark on a phase 3 trial. And so the whole idea is that from academic institution, your basic research and discoveries or innovations can also do some new guidance or new mechanism by which you can treat diseases which are, particularly in this case, it is which is a life-threatening situation, significant unmet medical need. And this will be a significant advancement once it is launched and we have post-marketing surveillance and we find them. So thank you very much, and that's the end of my presentation.
Thank you, Professor Anil, for that informative presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, Professor Gulati will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.